guys, I just wanted to take a few minutes and go through and explain in a little bit more detail some of the concepts that we talked about in today's lecture. So again, we talked about some gluten issues, and then we talked about Alzheimer's disease. And as I mentioned, this is going to be material that you will be responsible for on the take-home portion of your exam, but wanted to go through in a little bit more detail here. So we began talking about gluten, and so again, I want to kind of highlight what is gluten. So gluten is really a class of proteins that's found in four main food products: wheat, barley, rye, and spelt. About 75 to 80% of all the proteins in bread are actually gluten. And gluten really can be thought of as two proteins here, glutenin and gliadin. And they're actually linked by disulfide bonds. And so these two proteins actually present a problem for humans in their digestive tract. Specifically, gliadin is something that humans cannot digest. So again, wanted to take a second and go through kind of the normal digestion process. To highlight over here, if you'd like a little bit more detail about this, there's a video that's on Edpuzzle that you can look at that kind of goes through the details of protein digestion. But to just kind of summarize, proteins begin again in a folded state. Once we get into the stomach, the pH of the stomach as well as other conditions allow that protein to be denatured. And remember, we talked about the difference between denaturation and degradation. So denaturation is elimination of secondary, tertiary, and quaternary structure that still allows proteins to be connected via their peptide bonds. But then in the small intestine, we're going to eliminate that primary structure by having degradative enzymes that allow us to snip these proteins down into smaller peptides and, importantly, individual amino acids. And those amino acids are really important because only those amino acids are able to pass through the channels that we have within these intestinal epithelial cells. So again, remember, these intestinal epithelial cells are very tight with their neighbor. They represent a single cell layer in your intestinal tract. And this tight junction is really important. We'll get to that a little bit more on the next slide. So we use our protein channels here as a way to allow only fully degraded proteins as amino acids, carbohydrates as simple monosaccharides, and then lipids as triglycerides to pass through into our bloodstream. So just a little bit more history on gluten. Why is gluten useful? Why has gluten become an important staple in kind of our food service? And that's because the flour that we use in bread making and pasta making, once that's kneaded, that process of kneading the flour actually takes the gluten and allows it to agglomerate, which means it allows those gluten proteins to stick together. And that's what gives the elasticity to dough, that doughiness, that chewiness of it. So again, when we have these substances that are within bread and then we allow bread to rise, that chemistry is handled by yeast. And so we'll get to this in a few chapters once we talk about glycolysis. But when you take glucose and you break it down anaerobically by yeast for fuel, you go through a fermentation process that allows you to generate carbon dioxide. That carbon dioxide is what allows the dough to rise, and because of that elasticity, those bubbles become trapped. So again, that elasticity allows for bubbles to become trapped, and you have this nice, airy, bubbly bread that we have. So again, just a little bit of history with gluten, but we're going to break apart this concept of what your intestinal barrier really looks like on the next slide. So again, when we think about normal gut permeability, so these represent two intestinal epithelial cells. Remember that there's only a single cell that separates you from the external world. And so again, this is the external world, in essence, your intestinal lumen, the space that's inside your intestinal tract. And this represents the tight junction, which is a physical connection between intestinal cells. And sort of the status quo for this, what's kind of normal, is that the tight junction is tight and it's closed. And that allows the outside stuff to stay out, the inside stuff to stay in. And as we kind of highlighted on our previous slide there, the way that things get from the outside to the inside is generally by passage through channels that are within each intestinal epithelial cell. So, but then we'll ask the question, clearly these intestinal tight junctions serve some sort of purpose, especially if they could be opened. And so why or when would you open this tight junction? So the first is that maybe you need to let something foreign in and allow the immune system to deal with it. And so we'll talk about that on our next slide. But also, you might need to let large volumes of water out to deal with something foreign. And this is, again, when you have diarrhea, that actually serves an important purpose in flushing out your intestinal tract to try to rid that area of a foreign substance. So let's kind of delve in here now then to what happens when you have gluten that is inside your intestinal tract. So the problem is we mentioned before, no human can fully digest gluten. So basically inside of your intestines, it's not going to be converted into free amino acids. It'll become shorter peptide portions or completely indigested. And so it becomes a foreign antigen that lives in your intestinal tract. And so what we know happens with foreign antigens is these antigens are going to bind to receptors on the cell surface. You guys don't need to know the details of this slide, but you do need to know that there's antigen recognition on the cell surface of your intestinal epithelial cells. What's going to happen is it's going to increase the production of something called zonulin. And so zonulin is a protein. And so what this protein does is it opens the tight junctions. So again, zonulin is going to come up here. You don't need to worry about how it happens, but it binds to another external receptor and it stimulates the release of this tight junction. So we open up these tight junctions, and then this allows, again, the antigens to come in and interact with components in our immune system. So they interact with T cells here, and it's going to launch an antibody response. So then the question kind of becomes, well, what's the problem with your immune system seeing gluten as foreign? So again, if you're interested in some more details about how zonulin works and kind of some of the primary literature behind this, there's a video, again, up on Edpuzzle with how zonulin works. So why would gluten in your bloodstream be problematic? Why would an immune system response to gluten be problematic? And this kind of brings us to this concept of autoimmunity and molecular mimicry. So again, now foreign proteins enter your bloodstream and they generate antibodies. The problem is that they can then find and attack similar proteins in your body. So there's a short video up on Edpuzzle about molecular mimicry, but the concept is if you have an invading protein that is foreign, maybe it's gluten or maybe it's from some other foreign antigen that has a certain amino acid sequence that is the same as an amino acid sequence found on a tissue in your body, that allows your immune system to recognize both of these, to essentially not be able to distinguish between these, and you can launch an autoimmune response on that tissue in your body. So again, if a foreign antigen gets in and you launch an antibody response to it, depending on what that invading protein has mimicry to, has similarity to, will determine kind of what you attack. So if there's similarity to your cartilage, you can develop rheumatoid arthritis. If there's similarity to thyroid, and particularly for people who have Hashimoto's thyroiditis, which is the kind of downstream autoimmune effect to attack on your thyroid, wheat and gluten and dairy proteins casein are particularly important for people with Hashimoto's. Interact with tissues on your pancreas, you'll develop type 1 diabetes. Interact with the myelin sheath on your neuronal cells, and you can develop multiple sclerosis. Interact with tissues on your adrenal glands, and you can develop Cushing's disease. So what you'll see here is there's just a lot of different ways that gluten can, by launching an immune response, lead to several autoimmune diseases. So what should you think about with regards to gluten? And again, there's a lot of evidence that's out there that suggests that gluten can be problematic. A couple of videos that we kind of highlighted in class. There was a nice TED Talk that talked about, the fact that there's lots of primary literature out there suggesting that nobody can digest gluten. And so everybody is subject to this problem. There's a great TED Ed video talking about what is gluten. And so forth. So some things to think about is whether or not you are someone who is having a problem with gluten sensitivity, or as this video kind of highlights, maybe we call it wheat intolerance. One great way to kind of investigate that, it's not necessarily fun, is to undergo an elimination diet. An elimination diet basically is going to be about a 6-week period where you completely eliminate a food group from your diet. And maybe a best way to do this is to kind of eliminate everything. So eliminate all grains, eliminate all dairy, eliminate all sugar, eliminate any of these offending agents, and then let your body rest for about 6 weeks. And then you can try adding things back in and see if you have issues with them. And so that's a great way to kind of check as to whether or not gluten sensitivity is something you might want to consider.